Imagine you're building a house. You start with a bunch of identical red bricks. Individually, not that interesting, but start connecting them brick by brick, and suddenly you've got walls, then rooms, then a whole building. Now think of amino acids as the bricks of biology. They're small, simple, but when linked together, they form dipeptides, tripeptides, and eventually polypeptides, which are long chains that fold into functional living proteins. And just like the way you layer bricks determine if you build a cottage or a skyscraper, the order and shape of amino acids determine if you get muscle proteins, insulins, or a digestive enzyme. So today, we're gonna see how the bricks link up, how the walls form, and how the final protein structure is basically the blueprint of how life functions. Because in biochemistry, it's not just what you build, it's how you build it.
Another day, another sleigh here at Catalyst Learning Center. For today's biochemistry basics, we're going to delve into the world of dipeptides, tripeptides, and polypeptides. By the end of this lesson, you'll be able to define them, explain how they form, draw their structures, and even get to know real-life examples. So, what are we waiting for? Let's get straight into it. Okay, so let's rewind for a bit. What even are amino acids? Before we build anything, we need our bricks. And in biochemistry, amino acids are the bricks. Each amino acid has an amino group on one side, a carboxyl group on the other, and a unique R group that gives its personality. There are 20 standard amino acids, each having their own unique structure, and they're like Lego bricks for building proteins. So, how do we go from single amino acids to something useful? Here comes the peptide bond, the ultimate biochemical handshake. Here's how that works. One amino acid's amino group meets another carboxyl group, and they're like, hey, let's kick out a water molecule and link up. That's called a dehydration synthesis or a condensation reaction. So basically, lose water, gain a bond. Congratulations, you just made a dipeptide bond. A dipeptide is made of two amino acids linked by a single peptide bond. And when amino acids become peptides, they are given names. So, how exactly do we name these? Well, amino acids are almost always named from left to right. That is from the amino acid at the N terminal to the amino acid at the C terminal. We identify the left amino acid, change the "ene" of its name to "il", then connect it to the name of the following amino acid. Take glycine and alanine as an example. When glycine's amino group meets alanine's carboxyl group, voila, glycylalanine. What about valine and leucine? Can you name this dipeptide? If you answered valyl leucine, congratulations! Give yourself a round of applause. But wait, aren't there other ways to name them? Well, technically yes. You might see 3-letter or 1-letter codes used in protein sequencing, like GLIL and ALA, but these are usually used for longer chains. For now, we're sticking to the basic naming rules. You might ask, are dipeptides even used in real life? Ever heard of carnosine? It's a dipeptide bond found in muscles and brain tissues that has antioxidants and anti-inflammatory properties. But that's not all. There are many more essential dipeptides in this world that deserves more recognition. Okay, let's add one more amino acid to the party. Now, we've got a tripeptide, basically just 3 amino acids and 2 peptide bonds. So, how would we name it? Simple, we just follow the same rules. Let's say it's glycine, alanine, and valine. You go left to right, we start with the N terminal end, Glycine becomes glycyl, alanine becomes alanyl. Last one, we keep it as a valine. Therefore, we would get a glycyl alanyl valine. You may ask, why not just call it glycine, alanine, and valine? Well, because in peptide naming, we turn all but the last amino acid into their ill forms to show that they're bond in the chain. Do tripeptides even matter? Well, take glutathione. It's literally a detox superhero that protects your cells from damage. It fights off free radicals and helps protect your cells from oxidative damage. So yeah, tripeptides do matter a lot. Now imagine stringing together a 50, 100, even 300 amino acids. You just made a polypeptide. And now we're entering protein territory. But wait, the structure matters, so let's break it down. Primary structure. It's the exact sequence of amino acids, like beads on a string or letters in a sentence. One change in order, boom! Different peptide, different meaning. Secondary structure. Now we're folding. Some parts twist into coils called alpha helices. Others flatten into pleated sheets. The beta sheets, like origami, but molecular. But they can also fold randomly, forming random coils. These shapes are held together by hydrogen bonds. And yes, they're kind of fragile. Tertiary structure. This is the final form, the overall folding of a single polypeptide chain. The whole chain folds into a complex 3D shape, and this shape decides what the protein actually does. It's not just pretty, it's powerful. So imagine enzymes, antibodies, hormones, they all rely on perfect folding to work, which is quite neat if you ask me. But wait, there's more. When more than one polypeptide chains combine, they form the quaternary structure. But not all proteins form these. Remember, structure equals function. Mess up the folding, a protein might clump, stop working, or even cause disease. Think sickle cell anemia. Just one amino acid substitution and the whole structure changes. But when the folding goes right, proteins can digest food, carry oxygen, kill viruses, or even make your hair shiny. Basically, proteins are just chains. They're tiny origami machines, and their shape is everything. Let's try to sum these all out. Here's glycine. Here's alanine. Water's gone, peptide bonds form. And we have a dipeptide. Repeat and add valine. We form a tripeptide. Repeat and add several more amino acids. We eventually form a polypeptide. Easy, right? Now let's get to the exciting part. Since we already know how to identify amino acids and name peptides, let's do this little activity we have prepared.
Given the following amino acid sequences, draw the full structure of the resulting peptide. Show the peptide bonds clearly and indicate the N-terminal and C-terminal ends. We'll give you time, but feel free to pause the video if you need more. Time's up! Here are the answers. Did you get them all right?
Alright, here's a quick recap. Let's rewind and tie it all together. 2 amino acids walk up into a lab. Boom! You got a dipeptide. Toss in another one. Now we've got a tripeptide. Keep stacking. Welcome to the world of polypeptides. Remember, each amino acid has its own personality, like tiny characters in a protein drama. Thanks to their R group, it's like building with blocks, but every block has a personality. These guys aren't just floating around for fun. They show up in real life. Think muscle boosters like carnosine, or your bodyguard glutathione protecting your cells like a superhero. Peptides don't just chill. They fold, they twist, snap into shape. Primary structures, the exact order of amino acids. One change can mess things up. Secondary, curls and folds, local folding into alpha helices or beta sheets, held together by hydrogen bonds.
Tertiary structure. 3D drama, the full chain folds into a specific 3D shape, and this decides the function. Quaternary structure. Protein powers quad when multiple chains come together to work as one unit.
Before you leave, we'll leave you with an exit ticket just to check comprehension. No worries, you can answer these on your own. And that's it for our crash course. From bonds to folds to full on action, you've unlocked the secret life of proteins.